It's a pleasure to be here and speak to this group. This is a different group than I normally talk to. And so in preparing this, I was wondering what I was going to talk about. And so I'm going to try to keep it relatively straightforward and talk a little bit about how sorghum varieties are developed on a level that is rather simplistic, but I think it'll convey the information that you all need to know. And then also talk a little bit about how we can change things and modify the varieties that are available for farmers to produce, and how that might affect you as a buyer of that grain going forward in the future. And so my program as a background is at Texas A&M University. We're a public sector program, meaning that we develop material and we train graduate students in plant breeding. We use sorghum as the platform to do that while we're conducting research on the inheritance of traits that are important to the agronomic crop, whether that be grain or forage or other alternative uses that we might have for the crop. So we come at it a little bit differently. You won't go out to the farmer's field and see a Texas A&M hybrid growing, but you might see commercial hybrids that are being grown that are sold by commercial companies that very well might have material that was originated from our program. And so we tend to work behind the scenes on the research and education. And it's my goal here to give you a little perspective on how all of this interacts. So, to start with, we're talking about crop improvement or plant breeding, whichever one sounds more technically cool. You can use either one. The process is pretty simple conceptually. I teach plant breeding class, and this is the first slide I show in class, which will be tomorrow morning, so I'm getting practice tonight before I go there. It's basically 4 steps. You got to generate the genetic variation. We'll start with the red. Once we've got genetic variability, we want to select the best genotypes that do what we want them to do, whatever that may be. Then we have to prove that what we just selected actually works for the producer and for the end user, whoever they might be. And once we do that, we've got to make that available to the producer. And ultimately, if they adopt it, then you guys will buy it in the form of a grain product somewhere down the line. In all of these situations, you have to have agronomic performance for the producer or they won't grow it, and you have to have end-use quality for the end user or they won't buy it. And so there's multiple factors that go into all of these things for any crop. How we get to those is a function of several different things. First of all, what crop are we talking about? What is the biology of that crop? How fast does it grow? How do we propagate it, or how do we replicate it or produce it? And what part of it is important? Secondly, what technology does that crop have that you can use to improve it? Okay? If you want an example of that, corn has utilized GM technology for over 25 years now. We do not use GM technology in sorghum for a various set of reasons, but we do not utilize that technology, whereas they do. So certain technologies you see in certain crops, other technologies you don't. So it's a combination of putting that cycle together in terms of what does your crop look like, what are you trying to get out of it, and what technologies do you have available to do that. And it's the combination of those that define what can be done, and it's the economics in the markets that define what will be done. And so when we look at within the crop for that way, and then finally, I guess I'll add this with the little clip up here that came up, methods are always changing, and so we're constantly evaluating new technologies and saying, how does this make us faster, more efficient, or produce something that's better? And that'll always happen, but that goal will remain the same. So now specifically, we're going to talk about sorghum, and if you put sorghum in the context of what I just said, you guys have already heard tonight, or this morning, numerous times how wonderful the crop is, and how diverse it is, and how it grows in droughty environments, etc. So I'm going to keep that out of the discussion here, but I will tell you that when I look at it, I go, all right, what are we going to use it for? Well, we're going to use it for grain, forage, or bioenergy. That's what our program defines them as. Where are we going to grow it? Well, Texas has both subtropical production environments and temperate production environments, so adaptation of any material within Texas and across the producing areas will be important. It's in semi-arid regions, so drought tolerance is obviously going to be important. Now we get into some of the biology down here that interests plant breeders. We have a self-pollinated crop that we grow as a hybrid. A hybrid implies we cross two lines to make that crop. So the question is, how do you produce that hybrid if it is a self-pollinated crop, a crop that self-pollinates itself? Well, I'll talk about that in a minute. And then what kind of genetics are we working with? And suffice it to say that sorghum, luckily, we have relatively simple genetics, and we have some good genetic resources to work in that. And then finally, who are we working with? What kind of private industry do we have to do this with? Because the conveyor of the seed to the producer is the private industry. It's not folks like me in the public sector. We're a behind-the-scenes type group. So we need to be able to work with that private industry and within the context of their goals to make these things work as well. So here's that scheme again, except now I've fit it to sorghum, and you'll note a couple things in this schematic. First of all, that circle is still there. I'll point over there for y'all on the other side. But now there's two of them, all right? Why are there two of them? Why are there two of them there? Go back to that statement I said earlier. Sorghum is a hybrid crop. That means we have to have two parents to make a hybrid. One of them we'll refer to as the seed parent over here in this group. The other one is the male or the pollinator parent. And we have to keep them separate because we want to capitalize the concept of what's known as hybrid vigor. Hybrid vigor is the fact that a hybrid produced by two inbred lines outperforms either parent. And sorghum is a benefactor of that. If either one of those parents, let's say they would yield 100 units of something, a good hybrid should yield 150 to 175 units of that same product. That difference of 175 from 100 is the heterosis or the hybrid vigor that we have in that crop. Now we only get that if we keep these two groups separate so that we can capitalize on that heterosis. So instead of one circle that we have working in a sorghum breeding program, we actually have two circles that are kept separate from each other, and then they're evaluated for value through the progeny that you produce or the hybrid that you produce between those two groups. So that's within the context. And this applies to any of the sorghum types that I've mentioned previously. We're talking about grain primarily here, but it could be forage or energy or sweet sorghum or what have you that you're talking about from that standpoint. So how this works specifically, as you asked now, let's get to that question of a self-pollinated crop producing a hybrid. Okay? Remember that it is self-pollinated. What that means is that when you have a sorghum panicle that's growing and it's going through flowering, the technical term is anthesis, as it starts to flower, it has complete flowers. What that means, it has both the male and the female component present within each flower. And so when that anther or the male component sheds pollen, it typically will fertilize that ovule that's right there by it, all right? Now, if you want to make a hybrid, that's a problem because you have to move pollen from another parent onto the ovules that are there. Now, the way we do that in sorghum is a concept called cytoplasmic male sterility. It is a system in which we render a particular line male sterile by using a combination of genetic factors that inhibit the functional formation of pollen. So the ovule, the female part of the plant, is perfectly normal, but the pollen does not shed. It does not produce pollen, okay? So what we're going to do when we develop these new seed parents is we develop two lines, actually. The first line that's developed is called a B line, all right? And a B line is completely fertile, both female and male. But then what we do is we create another version of it called the A line, which has a different cytoplasm, and just take that to be a component of the cell that induces sterility within this line. These two lines are very similar because the only source of pollen for the A line is its identical version that's fertile called the B line. And so to produce the A line, you pollinate it with the B line. That recreates the A line to produce seed of the A line. And then the B line is self-fertile, so it pollinates itself and recreates itself. Now, that's how we produce the seed to produce the hybrid seed. When we're ready to make a hybrid, now remember back to our two circles, we had the pollinator lines over on the other side. Those are going to be the males. Well, we're going to bring them in here, and here I've called them the R lines, okay? So they're called the R lines. Those are the males. When we're ready to make seed of that hybrid, we take the A line, plant it in a block, and then we plant now the R line adjacent to it. The R line pollinates that A line in a picture of a seed production field like you see right down here, where the red is the female parent and the white is the pollinator parent. And then the seed's harvested off of that seed parent, put in a bag and sold to a producer. Now the difference between the R line and the B line is that the R line has the ability to restore fertility to that progeny, whereas the B line could not restore fertility. It maintained the sterility. The R line restores the fertility. That's why we call it an R line, okay? So when the farmer grows this seed right here and produces the crop that produces the grain that you buy, that hybrid is fertile because he used an R line to produce it. The other thing about the R line compared to that A line is they are genetically different. Remember our two pools? That's where we capture our hybrid vigor again, okay? So that's how the seed is produced, that is grown, that you get your grain from. It's also why you don't want to save that seed that's grown on the farmer's field and replant it again, because it will be a genetic anomaly. It'll be just all over the place. It'll be talls, shorts, reds, whites, all kinds of different colors if you were to grow that. But it looks uniform in the sorghum field because it's an F1. So, sorghum programs will have both seed parent development program, pollinator parent development programs. We have to have that cytoplasmic male sterility, all right? And breeding programs, typically, most of the programs that are out there today develop parents on both sides. But you're going to see that they tend to spend more effort on the male side of things because it's faster and easier. And I'll show you why in a minute. So let's talk a little bit about the process of development, because we've showed you structurally how that works. Now let's talk a little bit more in detail of timeline and what it requires. If you were to tell me you wanted a brand new hybrid that had a specific trait in it today that we had not heard of, no one had worked on, but you have a bag of seed that has this trait in it, and I want you to put it into a high-yielding sorghum hybrid, it's going to take at least 8 to 12 years. And it's going to take that amount of time and vary in that amount of time because of logistics of what is the trait, how hard is it to move into elite material, do I have to have it on both sides of the parentage, okay, both the male and the female, to get it to be expressed in the hybrid. And then I've got to test it once I do all of these. And oh, by the way, it needs to yield equal to or better than what's already out there for the farmer to grow, unless, of course, you're willing to pay a premium for it. And then it can be a little bit lower because they can offset that with the premium to justify the production. But just understand, these things don't happen tomorrow. It takes time to develop these hybrids. So when new hybrids are coming out that producers are growing and things change, it's because they were started 8 to 10 years ago, okay? And that timeline is still in play. We'll talk about how we can improve that, but the fact of the matter is this is not something that happens in a year. The other reason we have variable timelines up there is because, as I mentioned previously, people like to spend more time on pollinator parents because they are a shorter to market development period, typically minimum 8 years for those. If you talk about new seed parents, you're talking about 12 years to get to that, and I'll show you why in a minute for those things. The second component that's important here is the numbers that are evaluated. For every commercial hybrid that's out there, there were thousands, if not tens of thousands of hybrids that were evaluated for their potential and found to be not worthy of that, okay? And that's why you may hear, if you ever talk to plant breeders, that plant breeding is a numbers game. You have to look at a lot of material and use all the tools that you can find to find those perfect combinations that work and are adapted and have the end-use traits that people want. So for every hybrid that's developed, thousands to tens of thousands are evaluated during this process. Even if you have a perfect combination here, you have to look at the logistics of the process. And that, what I mean by that is you can have a wonderful inbred line that produces a beautiful hybrid, but if it doesn't, if you cannot produce that in a seed production area, too bad, because it doesn't work from that standpoint. So these logistics are important. Producer acceptance, this is one that I always get a kick out of because I oftentimes think that we put too many restrictions on what a phenotype of sorghum should look like. Is that the producer that's not accepting it, or is the marketer not accepting it? All questions that can be asked and debated, but can you change things, or will the farmer accept what you're sitting there? And then third, and just as important, will the end user accept it? Because we could improve yield in lots of different ways, but if the quality's not there and someone doesn't want it, it doesn't really matter. So with those things in mind, this schematic is a process by which a typical program would advance material through a breeding program. The schematic that's on here is for the pollinator lines, or the R lines, and what you'll see is that we will make what we call a breeding cross between two elite pollinator lines that may complement each other for a disease resistance trait and a quality trait, and maybe we want to extract something that has disease resistance and quality, or even better, we'll talk about sugarcane aphid resistance, has sugarcane aphid resistance and quality. And so we cross those two, and then we go through a series of self-pollination generations called the F1, 2, 3, and 4, where the plants, the genetics are segregating out and the lines are becoming what we would call inbred or uniform, phenotypically uniform. Most programs would go through the F4 generation, selecting through this process. Once they get to the F4 generation, and we've done very little, if no selection for yield, because remember, the performance of the inbred line for yield is not all that relevant, because it's the performance in a hybrid that defines whether or not it has value. And so we immediately start to evaluate these in hybrids as soon as possible. And in most programs, they will go through several phases of evaluations. Early phases where we're evaluating thousands of hybrids to identify the best combinations. Once you've identified the top performers from those thousands, you start to narrow that down, identifying the best inbred lines and the combinations that work best within those, until you get down to a few where you go to widespread testing and you start testing the logistics of seed production to be able to, can we commercially produce seed of this hybrid at a cost level that makes sense to sell it? And does the performance across a wide enough range of the sorghum production region, is it suitable so that we have a large enough marketable area to sell that hybrid within? And does it fit the maturity and the height and the disease reaction profiles for all of these things? And then finally, if it does those things, does it have acceptable end-use quality for someone to use? So that's the R line, and you can see the schematic on how that works. Now, this is the same slide, same words on the left side, except now, and I apologize for some of you folks that are translating, because these slides aren't in the packet. So you'll have to work through that. But this schematic shows for the B lines, the seed parents, and you'll know there's a lot more stuff on there. And this is actually picking up where it's the exact same. At this point, it was the exact same as what we saw in the R lines, so this is all additional for the B lines. What's happening here is that we have to make a male sterile version of that line because in the F4, it was male fertile. So these steps that you're seeing here are related to producing that A line that I talked about earlier. How do you do that? Well, we have to do a technique called backcrossing in order to convert that B line up here into an A line that then we can do hybrid testing with. Because remember, before that, we can't really make the hybrid because we don't have a sterile version to make a hybrid with. So what that means is that it takes more time, additional 4 to 5 years, to get all of that done and then start testing. That's why folks in the industry are very reluctant to change the seed parents that they use because it just takes a lot more time and effort. And the stringencies on quality are much higher. It cannot break sterility. If it breaks sterility, you no longer have a hybrid, you have a mix of things, and nobody wants to buy that. So these traits are really, really important in these seed inbreds, and it's very meticulous work, and that's why it takes longer, and that's why they don't often like to change them because of the challenges associated with them. So what I thought I'd do next is that's the process in a nutshell. And that sounds probably not simple, but the process takes time, it takes numbers, and not all that takes money and resources to do that. So the question becomes, if you're going to, and I haven't even talked about traits, I'm going to talk about those at the end here. If you want to improve this breeding process, you look at new technologies to do that. And I've broken these up, and I'll hit on a few of them. I don't have any more slides about this, but just talk about the concept of how we could improve the breeding process. Improving the breeding process in this development program requires you to do one of several things. Speed it up, all right? Can you make it go faster, all right? Can we go through that process I described that takes 8 to 12 years? Can we do that in 5 to 7, all right? And if we can, how do you do that? Secondly, improve efficiency. Improving efficiency means that we measure things faster, or we evaluate, because we're more efficient, we can evaluate more material more effectively within the process of doing these things. And then the third thing is the traits. Can we put something in there that peaks performance either for the producer or the end user in terms of traits? So what I thought I'd do is just spend a few minutes on each of these and explain what I mean by them. All of these that are listed up here are things that are happening, have already happened, or are in the process of happening in some way, shape, or form. All right? Speeding the breeding process. Well, there's several different ways that are being tested and evaluated in sorghum right now. I'm going to start with the bottom one. I put marker-assisted selection on here, / add to that genomic selection. What this is using genetic technology, genotyping technology, where we can go in and assay all of the chromosomes in that plant species and know their allelic composition. That's what version of the gene they have throughout that whole genome, throughout that chromosome, and then associate that back with a trait of interest. A trait of interest in this case would be yield, okay? Now we're not trying to find where these genes are located when we're doing genomic selection. We're just simply trying to say, if a plant has this genomic constitution, it has the predictive ability to yield this much. And what we can do with that is if we have this technology in place where we can take a new inbred line, genotype it, and predict the yield, we can look at more material because we don't have to look at it in the field. We can look at it in the lab. We can look at it in the lab and go, oh, we just looked at 100,000 things in the lab, and these are the top 10,000, okay? So now, maybe my breeding program, I can only evaluate 10,000 new hybrids a year. But if I could genotype and evaluate 100,000 and cherry pick from that 100,000 the top 10,000 to put in my program, I just increase the numbers that I can look at. And I've put a higher priority or a prioritization on these are the most likely to be the highest yielding, okay? So genomic selection is one, marker-assisted selection is a form of that as well, that has the potential to speed the process and really also increase the efficiency of the programs as we go through that by increasing our numbers. Haploid induction. In the corn world, they use what's called doubled haploids. You saw those 4 steps of inbreeding that I described. They do that in 2 steps, okay? By creating a version of a corn plant that has only, sorghum and corn both have 20 chromosomes in their natural state. They create a version that only has 10, and then they double that to back to 20. And by doing that, they create an instant inbred line. Remember, that's what we talked about. We do the same thing in sorghum, except we have to self all the way through to do that. And that takes more generations. USCP funded research with Pioneer to identify lines that facilitate this haploid induction in sorghum, and they're being evaluated right now. If we are successful in deploying those have the real potential to increase the speed at which we go through that inbreeding process and reduce that cycle time that I talked about earlier. Gameticides. This is another one that if you're going to do haploid induction, you've got to have gameticides, and then we've found in other ways that gameticides are important. What is a gameticide? In this situation, we're saying specifically a male gameticide. It inhibits pollen formation, but leaves the female flower functional. So if you have a gameticide, it facilitates several things. First of all, you can use it in your haploid program. Second of all, it opens up the opportunity for testing of new bee lines before we go through that arduous steps of sterilization. If I know which lines are going to be the best in hybrids, then I only have to sterilize those lines to do that. And so that allows us to reduce the size of this sterilization process just down to select few lines, and allows us to look at more material earlier in the program within that context. Under improving efficiency, I have phenomics and remote sensing. You guys heard of UAVs? This is what we're talking about here. Can we phenotype more efficiently? Can we measure traits as my graduate students without a stick, okay? If you want to measure plant height, could you do that with a UAV? I'll submit to you that you can. If we look right here, these 2 pictures I've got down here and these up here are both examples of remote sensing work that our program's been involved. There's been numerous other programs doing this kind of work. From my perspective, from a breeding program, the question is, and it has lots of other applications, but from a breeding efficiency perspective, can we take an image like this and convert it using different software and packages and algorithms so that we can extract useful phenotypic information that we traditionally took on the ground? Specifically, things like plant height, days to maturity, grain yield, lodging. All of those are really important things for both a producer and a plant improvement program to know about these potential hybrids. If we can do that, it improves our efficiencies. For example, if I want to measure grain yield, I have 2 ways to do it. Well, 3, there's numerous ways to do it. One of them is to go out there and visually estimate. This one's going to yield more than that one. Don't have a number. I can't do statistics on that. A second way is to send the graduate students out there and tell them to hand harvest that, thresh it, and give me the number. They don't like that, all right? The third way is I can drag a combine all over the country and combine that and then report the numbers back. That takes time, effort, money, equipment, etc. If I can estimate those things and be relatively accurate, I can again expand early in the program, look at more numbers, find the best things, and then move into more detailed testings at later phases in the program. So it improves efficiency by allowing us to do more with less and also improve the chances of getting it right. And so we've been looking at these tools, as many other groups are as well, and how you actually deploy them within a breeding program to make things more efficient. When we talk about unique traits, I'm just going to hit on a few. What I'm talking about here is mostly traditionally identified traits. And what I mean by traditionally identified is that these are variants, I use the word variants rather than mutants, that were observed in the field, that people observed, found, discovered through natural variation development, okay, through mutation, etc., not through systematic or not through GM technology. And you'll note down here, there are no commercially available transgenic sorghum hybrids. Didn't say there's not any transgenic sorghum, there's just none that's been commercialized and probably won't for quite a while. For many different reasons. But what we're talking about are traits that have been identified within the material that have value and can be treated as a trait. Specifically, herbicide resistance has appeal to producers, all right, because it allows them to bring those things. Forage quality, because it improves quality, makes it more appealing for a dairy to utilize, etc., if they wanted to. More recently, you've seen things such as specialty grains, and we're going to talk a little bit about that on the next slide. And some of the folks in here are interested in some of those specialty grain traits as well that could be utilized within those. All of these to date have been developed through traditional selection and identification within the sorghum germplasm that exists writ large. It does not mean that we couldn't do gene editing. In fact, there are numerous groups that are actively engaged in talking and doing gene editing of targeted traits to modify the genome or the gene so that you get a desirable outcome for that specific trait, whether it be an agronomic trait, whether it be a quality trait, or something else that I have unforeseen or don't know about that's out there. When it comes to gene editing, a couple things that y'all should know. First of all, not all gene editing is going to contain, some gene editing results may contain genetic modifications or GM traits in them. Others may not contain those at all. What I mean by that is it's possible for someone to gene edit a trait, and there would be no way for anyone to know that it was gene edited unless they told you that. Okay, because of the way there's no evidence of any genetic modification that occurred in that process. It's been eliminated through the selection process. And so it looks completely, and it is, a completely natural edit that occurred just in a more systematic and targeted way. Currently, as I understand it, in those situations, those would not be considered GM crops in the crops that are using that right now. So all of these things are available. Let's talk a little bit more about traits. I hit on these. And this is traits of importance in general. I'm not talking about traits that are single gene traits, but these are the things that a program is going to find a priority on when they start talking about improving the crop. They're sort of in order of priority as I perceive it. Number one is yield. Probably number two, probably number three, and four are all yield, all right? But those other traits below yield are all related to yield, because if you don't have them, you won't have number one. Standability refers to the ability of that plant to stand and not fall over before it's combine harvested. So it's not lodged, okay? The other word for this is lodging. We don't want lodging. We want things to stand up so that they're easier to harvest by the producer. A package of pest resistance, biotic pest resistance, which can include resistance to disease, resistance to insects, herbicide tolerance packages. This is where some of the unique traits may come in if we get some of those. All of these things have value in varying levels depending on where the producer is and what the pests and climate situation is that's important to them. For example, in some places in South Texas, there's diseases called head smut and downy mildew that are very important to producers, and the best way to control them is through genetic resistance. So having that resistance in those hybrids grown in that area is very important. Whereas if you're in western Kansas, those diseases are not exactly a problem there, so they're not going to worry about the presence of resistance in a hybrid that they might grow. Abiotic stress tolerance, what I mean by that is drought tolerance, heat tolerance primarily. You can talk about some nutrient tolerances or tolerances to nutrient deficiencies here, but mostly we're talking about this one right here. The ability to withstand drought and periods of drought during the growing season. All right, and sorghum is known for that. That's why we still grow it in drier areas. However, there is wide variation within sorghum. Different sorghum genotypes react differently to drought stress. There's two specific types that we talk about in sorghum. One is called pre-flowering drought stress. That's droughts when a drought occurs before the plant enters the flowering phase of growth. And the second phase is called post-flowering drought stress, wherein the drought stress occurs after anthesis or flowering and continues to terminal growth or the end of the growing season. Of those two, post-flowering has always been the most important, but pre-flowering is also important, especially given changes in climate that we're encountering. Heat tolerance is another one. You could put cold tolerance up here. There's a lot of different things that people have been working on. But this one's the benchmark of that. And then the last one I'll talk about here with this group today is grain quality. And I'll just point out a couple things that are around and you guys are familiar with. Some of you are familiar with. Grain quality. The first one would be food quality, okay? For many, many years, sorghum was a feed grain in the United States. We fed animals with it. We still feed animals with it. But about 20 years ago, the concept of food quality sorghums first started showing up in the mid-90s. And that was defined as something that had white grain on what we call a tan plant. A tan plant is the base color of a plant. If you don't know what I'm talking about, use your tree analogy. When a tree turns colors in the fall, that's the basal plant color of that tree. It's not the chlorophyll, it's the base color of that tree, whether it be red or purple or yellow or what have you. Same thing in a sorghum plant, you have a plant color, which is usually either pigmented or non-pigmented, and a non-pigmented is commonly referred to as a tan plant. So we want a plant that has white grain and a tan plant. Both of those are important because it produces a grain that produces a white, clean flour with no color staining in it, etc. This was food sorghum defined for many years. You'll also note I said that concept became available with inbred lines capable of producing those types of hybrids in the early 1990s. It's been in the last 5 to 10 years that food sorghums have come into their own, and you see them on the shelf. Why did it take 20 years to get there? Go back to the example. It takes 8 to 12 years to develop new hybrids with the right adaptation and competitive ability to yield to make it into the marketplace. And so those things showed up at about the time that we got our real competitive hybrids out there as the private industry got a hold of them and made them into what they are today. In the meantime, we've seen what food quality in sorghum means change a little bit and add some new things. They've already been mentioned a little bit today. Specialty sorghums, high antioxidant, high tannin, these two together. This is a black sorghum. I saw some slides here that had black sorghum in them. Well, this is a black sorghum hybrid that our program developed and licensed and is an core ingredient in this grainberry cereal, and it's sold specifically for this antioxidant and high tannin capacity. So those markets do exist. They're niche markets, but they do demonstrate that these things have come a long way in the past 10 years and people are utilizing them. More recently, you see interest in waxy endosperm hybrids. You see interest in high protein content and high protein digestibility for feed and food uses. I'm happy to report to you that we have these types, but are they in commercially available hybrids right now? Not really, okay? How long is it going to take for them to get in there? Well, they're starting to come, but it takes time, okay? And so they have to be developed into these backgrounds. Now, can we use some of those tools to do this more efficiently and faster? Very possibly. So are we looking at 15 years to make this happen? Probably not. But some of that is defined by how motivated the end users are to get these materials, because people can produce these hybrids faster, but there's going to be a trade-off for them, because usually if you're developing, go back to that example I stated earlier. If you have a new trait and you want it in a new hybrid, that's competitive, it's going to take 10 to 15 years to do that. If you want one faster, the trade-off is yield. It's not going to yield as high, and so to get that grown, it's going to require more of a premium to make that happen. So all those are trade-offs in this concept of that. Our program has worked in these areas, just as a side, for many years. Like we already showed you this example, we're testing waxy hybrids now from different programs, and that's sort of the role, quite frankly, I view as public sector, is that if we're not out in front of this, we're probably not doing the right thing, because that's what we should be doing, because we're not trying to sell a hybrid right here today and now. Here's the gloom color differences. This is why we want a white tan grain, because you see right here is a white tan. This is a white grain too, but it has pigmented glooms on it, and when you make flour out of that, it has a discoloration to it that people do not like, whereas this makes a nice white flour. This schematic has a waxy endosperm versus a normal endosperm, where a normal has 75% amylopectin, 25% amylose, approximately. There's variation. And waxies get close to 100% amylopectin. They've still got some amylose in them. I'll finish up with a summary of what I just told you. New hybrid development takes time, all right? And I won't say that, and the variation depends on what it is that you want, how bad you want it, and what form you want it, all right? New traits in sorghum are possible, regardless of the way we get that new trait, whether it be through traditional phenotypic screening of material to identify lines that have it, or a gene editing event, or even go so far to say if someday GMs are accepted and viable in sorghum through a GM event. Regardless of how we get that, it still requires that incorporation. Just because you have the trait in something doesn't mean that it's commercially viable to be grown anytime soon. So once you see this, go back to point one, all right? Once you have it, you've still got to do the development on that, all right? Whatever we're going to do, we've got to have value to someone, preferably to both. And I've just broken up producer and end user, but there has to be someone that's going to pull this through. I learned a long time ago that just saying I like something got nothing out of it, all right? There had to be somebody else that was an advocate and was willing to pay for it, all right? Identity preservation, IP and grain, I don't think that's a big deal anymore, but it is probably something that's very important. And again, I'll reiterate that to date, we don't have any GM sorghums out there that you have to worry about.